Hi everyone, welcome back to class. Good day, everyone. Today, what we want to talk about is the Krebs cycle, also known as the citric acid cycle, also known as the tricarboxylic acid cycle. So, in short, TCA. That is what we want to discuss today. It's a metabolic pathway in biochemistry and is one out of numerous metabolic pathways that we have in biochemistry. And you know how we do things here? We break things down in simple English so that everyone can learn. We will discuss this pathway. We would look at the intermediates that are all present in the pathway. We look at the enzymes present in those pathways, and we will look at the functions, the usefulness. Why do we even need this pathway in the first place? And lastly, we will now look at shortcuts, short forms, tricks, and tips that will help us to remember this pathway anytime, any day. First, what is TCH cycle? It's a metabolic pathway that helps to break down acetyl CoA, and the aim of breaking down this molecule is just to give us energy. What TCA is all about is that it collects acetyl CoA, then work on it, break it down to give the cell energy that is needed. That's just a summary of everything about the TCA cycle. And in our previous class, in one of our previous classes, we talked about the integration of metabolic pathways, where we looked at it in respect to carbohydrate metabolism, protein metabolism, and fat metabolism. And in all these metabolism, we said all of them will give us acetyl CoA, and this acetyl CoA will now enters into the TCA cycle. So we can say this class is a continuation of that class. For those of us that have not checked out that class, I will put the link in the description so that you can check out the class and you can follow us very well. So when we get this acetyl CoA and it enters into the TCA cycle, the TCA cycle will now help us to break this acetyl CoA down, which will ultimately give us energy. Now let's look at the pathway itself. We've discussed that one, the acetyl CoA that enters into this pathway, that serves as the starting point for this pathway, can be gotten from different sources. It can be gotten from carbohydrate metabolism through glycolysis, and which will give us pyruvate, and the pyruvate will now be converted to acetyl CoA through the pyruvate dehydrogenase complex that we said it has like 5 enzymes in that complex. Now, the acetyl CoA can also be gotten from fat metabolism through the process of beta oxidation. That will give us acetyl CoA. Then also from the metabolism of protein through different processes, including transamination, we can also get acetyl CoA. Now, we already have our acetyl CoA. That is where we are starting this class from now. We have our acetyl CoA. This acetyl CoA will react with oxaloacetate. So, so the reaction of acetyl CoA with oxaloacetate will give us citrate. This reaction is catalyzed by the enzyme citrate synthase. Remember that we said in when we were talking about metabolism, the different metabolism, different pathways in a different metabolic pathways in biochemistry. We said all these reactions are being catalyzed by enzymes. So every metabolic pathway, every reaction that occurs in biochemistry, mainly they are catalyzed by enzymes. So the enzyme that catalyze this reaction, catalyze this reaction to give us citrate is citrate synthase. Upon getting this citrate, aconitase enzyme will help us to convert this citrate to isocitrate. So we have citrates to isocitrates, and the enzyme that catalyze that is aconitase. Isocitrates will then be converted to alpha ketoglutarate, and the enzyme that catalyzes that reaction is isocitrate dehydrogenase. Here, there will be production of electron carrier. NAD plus is being used up to now give us NADH plus H plus. Now, let's move on. Alpha ketoglutarate will now be converted to succinyl CoA, and the enzyme that helps us to catalyze that reaction is alpha ketoglutarate dehydrogenase. And in this reaction, there will also be production of another electron carrier. Succinyl CoA from this reaction will further be converted to succinate. And the enzyme that helps us to catalyze that reaction is succinyl CoA synthetase. In this reaction also, there is production of GTP. One molecule of GTP is produced from this reaction, which is succinyl CoA to succinate. Moving on, our succinate will be further converted to fumarate, and the enzyme that will help us to catalyze that reaction is succinate dehydrogenase. And in this reaction also, there is production of another electron carrier, giving us FADH2. Fumarate will now be converted to malate, and the enzyme that will help us to catalyze that reaction is fumarase. Our malate will now be converted back to where we started from, which is oxaloacetate. So our malate will be converted back to oxaloacetate, and the enzyme that will catalyze this reaction is the enzyme malate dehydrogenase. And in this reaction also, there is production of another electron carrier. So NAD plus is used up to now give us NADH. So after getting our oxaloacetate, the cycle can now continue to revolve. Now let's have a broad overview of this metabolic pathway. Now we said from acetyl CoA reacting with oxaloacetate to give us citrate, and the enzyme that catalyzes that reaction is citrate synthase. Now citrate will give us isocitrate, and with the help of the enzyme aconitate. Isocitrate will give us alpha ketoglutarate with the help of the enzyme isocitrate dehydrogenase. And in this reaction, NAD plus is converted to NADH and hydrogen ion. Also, alpha ketoglutarate is now converted to succinyl CoA with the help of our enzyme alpha ketoglutarate dehydrogenase. Also here, electron carrier is being produced. From succinyl CoA is now being converted to succinate. The enzyme that catalyzes that reaction is succinyl CoA synthetase, and here GTP is produced. Moving on, succinate is now converted to fumarate with the help of our enzyme succinate dehydrogenase, giving us another electron carrier, which is NADH. Then fumarate gives us malate with the help of our enzyme fumarase. Malate is now converted back to oxaloacetate, where we started from, and the enzyme that catalyzes that reaction is malate dehydrogenase. And here also another electron carrier is being produced. Moving on now, let's look at the tricks that you can use to remember everything about this pathway. Let's start with the intermediates of the pathway. What I mean by that is oxaloacetate, citrate, isocitrate, alpha ketoglutarate. Those are the intermediates of this pathway. Now let's look at them. Officer, can I keep someone's spare for money? Now let's break it down. Now, officer, this means oxaloacetate. That O stands for oxaloacetate. Can the C here stand for citrate? I, the I here stands for isocitrates. Keep the K there start for alpha ketoglutarate. Someone, the S here stand for succinyl CoA. Special, the second S stand for succinate. For the F there stand for fumarate. Morning, the M there stand for malate. Then we come back to this officer which stand for oxaloacetate. So when next you want to remember your TCA circle very well, just remember this. Officer, can I keep someone special for morning? So when you first write that down in your book, then now be using the first letter of each words to remember your TCA. Let's now move on to the enzymes that are in this pathway. How can we know these enzymes? What are the tricks in knowing these enzymes? Let's move into that. Now, this is what we want to use to remember our enzymes that are present in this TCA circle, the way we did for all the intermediates that are there. So let's try to pick them one after the other. Let's start with the CS. The CS stands for the citrate synthase. Then the A here stands for aconitis. The IS stands for isocitrate dehydrogenase. The K here stands for alpha ketoglutarate dehydrogenase. The S here stands for succinyl CoA synthesis. Then the S here stands for succinate dehydrogenase. The F for the focus there stand for fumarase, while the M there stand for malate dehydrogenase. Now, let's check this. In the reaction involving the conversion of acetyl CoA plus oxaloacetate to citrates, then we have one molecule of water there, H2O. That's one. In the reaction that involved the conversion of isocitrate to alpha ketoglutarate, there is one molecule of CO2 there. Also, the conversion of alpha ketoglutarate to succinyl CoA, there is also the release of one molecule of CO2. In the reaction that involves the conversion of fumarate to malate, it gives us another molecule of H2O. Also, in the reaction that involves the conversion of succinyl CoA to succinate, we have one molecule of GTP. So, in total, in general, for this TCA circle, we can now say that the direct products of these metabolic pathways are one, two molecules of H2O, then two molecules of CO2, then one molecule of GTP. Now, other product that we can get from this place are the electron carriers. So, from this pathway now, the electron carriers that we have there, one, at this isocitrate dehydrogenase. So we have one electron carrier there. At this alpha ketoglutarate dehydrogenase, we have another electron carrier there, making two electron carriers. Another dehydrogenase, at this succinate dehydrogenase, we have another electron carrier there, making it three. Then at this malate dehydrogenase, we have another electron carrier there, making it four. So in total, we have four electron carriers that we could get from this pathway, which include one molecule of NADH2 and three molecules of NADH. So let's write it there. 3 NADH, then one FADH2. They will now move into the electron transport chain. That is another metabolic pathway. So when it now move there, that is where it will now be used up to produce ATP that the cells need to carry out every activities in it. I hope by now we understand everything now about the Krebs cycle, which is also known as the citric acid cycle, which is also known as the tricarboxylic acid cycle. Thus far in this class, we've looked at the TCA cycle. We look at all the intermediates, the enzymes, then the products, and we also included in our class tips for us to know each of these intermediates. We said for the TCA intermediate, we can use officer can I keep someone special for money to know each of these intermediates there. And for the enzymes of this pathway, we can use this. She has always insist keep special should focus more. We can use it for this in remembering this the enzyme involved in this pathway. And another thing for you to remember where all your electron carriers are, just know that anywhere you see that dehydrogenase, be it malate dehydrogenase, be it the associated dehydrogenase, well, just for this pathway, just know that anywhere I'm saying that dehydrogenase, bear it at the back of your mind that there is always production of electron carriers there. So, and we in total we said we have three NADH, which are electron carriers, then one FADH2, which is also an electron carrier that will now be transferred into the electron transport chain, where it will now be converted into ATP. With this, we've come to the end of today's class, and in our next class, we'll try and look at how this pathway could be regulated, places where it needs to be inhibited, where it needs to be activated, and why do we actually need to regulate this pathway. These and many more is what we're going to study in our next class. See you in the next class. Bye bye.